DNA microarrays provide the means to analyze patterns of gene expression at different time points in a living cell, a technique developed by Pat Brown, where cDNAs can be embedded onto glass slides. Using these DNA arrays, he could do large-scale expression studies. Growth stage-specific mRNAs are isolated and then reverse transcribed to give unique cDNA populations. These are directly embedded onto specially coated glass slides. The slides are coated with polylysine, which is positively charged. DNA is negatively charged, so the cDNA sticks to the slide through an ionic interaction. The cDNA can still interact with a DNA probe. Brown used arrays to obtain gene expression profiles of cancer. Diffuse large B-cell lymphoma, DLBCL, is a common lymphoma, cancer of the lymph nodes. He found subtypes of DLBCL that correlated with survival rates. Brown made a chip with genes expressed by the lymph nodes and those important in cancer biology. A total of 17,856 cDNA genes were printed onto what we called lymphochip. Each square on the chip corresponds to a different cDNA. Then, he made cDNAs from different DLBCL tumors. He labeled one set of cDNA with a red fluorescent tag, the other with green tag. Brown incubated the arrays with the tagged cDNAs, which bound to the matching genes printed on the array. Since he knew the positions of the genes on the DNA array, Brown could figure out the levels of gene expression based on the color signal. If the gene was only expressed in DLBCL1 cells, the square was red. Similarly, if the gene was only expressed in DLBCL2 cells, the square was green. If the gene was expressed equally in both cells, the square was yellow. Thus, he identified two subtypes of DLBCL. These subtypes have different responses to therapy, and with this type of diagnosis, more tailored treatment can begin for patients. This type of tailored treatment is called pharmacogenomics.